everyone to our monthly international webinar series of the Global Future Alliance. Today, I am delighted to introduce Dr. Danielle Delp. Danielle received her PhD here from the University of Maryland College Park under the advisement of Dr. Stephanie Lansing, and she was also one of our very early UMD Global Stewards Fellows. She was in our 2020 cohort, which was a bit of a challenging cohort, but it was so wonderful to have her as part of our program, which seems like many years ago now. Currently, Danielle is a postdoctoral associate researcher in the Environmental Science Department of Rutgers University in New Jersey. Her areas of research interest include bioremediation, anaerobic digestion, and environmental justice. Her research emphasizes the use of living things, such as algae, bacteria, and plants, to help clean up dangerous and persistent pollution in the environment, with an emphasis on the practicality and affordability of solutions to maximize accessibility to local communities. Her current research is using bacteria to mitigate contamination from river sediments on the Passaic River in Newark, New Jersey, where decades of chemical manufacturing and careless dumping have left heavy contamination. Today, Danielle will be presenting findings from her current research on the Passaic River, as well as findings from research she has collaborated on with the similarly contaminated Tittabawassee River, which I might not have pronounced correctly, downstream from Midland, Michigan. So again, welcome everyone, and welcome Dr. Delp.
Thank you for the lovely introduction, Amy. It's great to be back again, seeing all of you. I know I already know like half of you here, but it's lovely to see you all again. Thank you for inviting me. Today I'm going to be talking about the work I've been doing but in New Jersey here at Rutgers University and dealing with legacy pollution, things that have been in the environment and have been staying in the environment for years and years and years, specifically dioxins and furans. So my background, as Amy said, BS, MS, and PhD in environmental science all the way down. And I was the global steward in 2019, 2020. And I know Amy already mentioned my research interests, but bioremediation is kind of my cup of tea. It's a topic that's really important to me because it is an alternative approach to be cleaning up these really troublesome environmental pollution sites that we have in the US. And for me, it's especially a huge personal concern because I grew up across the street from a site like this. Like literally, my mom still lives in that house. Actually, her front yard faces it directly, and you can see this giant white fence, like maybe 50 meters away. It says, "Do not enter United States Superfund. You know, hazardous, dangerous, awful. Don't enter." So these types of hazardous sites are something I've grown up with my entire life and had to deal with the consequences of living near. And so I have a lot of personal interest in, well, one, cleaning them up because, a lot of these sites sit for decades without getting cleaned up. But doing it in an affordable way too, because money is kind of the be all end all of getting these projects done. Um, so to go into a bit about the Superfund sites, because that's what they're called. 'Cause I know not everybody here is environmental science. The National Priorities List, which is also just called Superfund colloquially, it was started in 1980 by the U.S. federal government in response to the Love Canal disaster in Buffalo or Niagara Falls, New York, in the 1970s. Basically, a chemical company or multiple chemical companies have just been dumping this chemical waste into an old dry canal, and then they just capped it and sold it when it was filled, and they built a school on top of it. And predictably, a lot of local children were exposed to chemicals, and it caused a massive cancer clusters and massive clusters of other diseases and disabilities and pretty horrible things. So basically, starting in 1980, the US government was like, all right, in response to a lot of lobbying by the victims of the disaster, we have to hold these companies accountable, we have to force them to clean it up, and we have to get these things remediated because people can't be living with this. So the priorities list is a registry of the most contaminated sites in the US. And that includes places that were contaminated by legal dumping before regulations existed, before the EPA, before rules were in place, as well as illegal dumping that happened in the past and still continues to happen, including by organized crime here in New Jersey. Um, Obviously, this is really bad for human health because these tend to be sites on cheap land, so industrial land, land near poor people, biracial minorities. It's a huge environmental justice issue on top of being a public health issue. And one of the complicated things with dealing with these sites is that each of them is contaminated with different things. So remediating them involves not just identifying like the history of the pollution at the site, which can go back decades or even a century or more. That's one of the sites we're going to be talking about today is. Um, but also you have to figure out, okay, what was dumped there? How has it changed over time? How's it moved around? So just characterizing what you've got, establishing a baseline is like years of work by itself. And these are some of the common contaminants, at least in the sites here in New Jersey. We got volatile organics, that's benzenes, TCE. That's also called PERC, it's dry cleaning fluid. Heavy metals, one of the sites in my hometown was heavy metal contamination. Organic pollutants like dioxins, furans, PCBs for the topic of today's talk, and a whole lot more. There's actually one famous one near here at Rutgers that it's where they dumped radioactive material from the Manhattan Project back in the 1940s. So lovely. And the reason why this is such a problem in New Jersey specifically is we have the highest number of Superfund sites of any state in the country, despite being the fifth smallest state. And this is also bad because we're the most densely populated state, which means highest amount of waste exposing the densest populated communities in the country. Because you can see we've got New York City, we've got Philadelphia, major population centers of the United States are being exposed to this stuff on a daily basis. So it's a real public health hazard here. So just talking now about the sites that I work on. The first one is the Diamond Alkali Superfund site in Newark, New Jersey, which is pictured on the right here. Um, this was a site of chemical manufacturing of myriad chemicals, but especially pesticides, starting in the 1940s, including lovely DDT, which is very famous for a whole suite of problems. But the one that's relevant to my research was the herbicide Agent Orange, which was used as a chemical weapon during the Vietnam War. And because basically, as they were manufacturing this stuff, they were dumping the residues directly into the river. Um, so it was identified for the National Priorities List in 1984, so it was one of the first sites added to the Superfund list. But the scale of the contamination is so huge, it's like they've cleaned up the hot spot of where the chemical factory used to be, but the river is still contaminated, like the whole river bottom. It's 17 miles of river. That's a huge cleanup problem. Logistically, financially, uh, you know, emotionally. And that the contaminated zone also includes parts of Newark Bay, Hackensack River, Arthur Kill, and Kill Kill, which is these little straits that are between New Jersey, Staten Island, and New York City, like Manhattan. So, a really populated area that is all contaminated with all of this horrible stuff. And similarly contaminated is the Tittabawassee River, which good try, Amy. You got almost on the ball there. The Saginaw River and Bay Superfund site in Michigan. So this is if you're familiar with the Great Lakes, it's a tributary of Lake Huron. This one goes back to 1897. It was it was and remains a site of one of Dow's major chemical factories. Over 1,000 chemicals produced and then 1,000 chemicals dropped in the river, probably. It's about 25 miles of contamination. It's about a little longer. Still pretty huge. But the problem with this one in particular, and this the picture on the right, is actually from during a major flood that happened a few years ago. Um, because of the way the river is, every time it floods, it's also contaminated the surrounding land, which includes farms, people's neighborhoods, commercial properties. It's a giant mess. Um, but it has a few of the major chemicals present are very similar to those present in the Passaic River contamination, which is why this project is paired together. So we have collaborators at Michigan State University that send us sediments from here. And so a lot of our research has also been on this river as well, using the same methods that we use for the Passaic. So this is the contaminant that's kind of the big hot button issue at both Tittabawassee and Passaic River. Dioxins and furans. So these have a billion different names depending on the paper that you're reading, the newspaper article. Sometimes they'll just say dioxin and furan. Sometimes it'll say PCDD, PCDF, which stands for polychlorinated dibenzo, whatever, as you can see. I also just see it as chlorinated dioxins furans and just dibenzodioxin dibenzofuran. They all are the same thing. And the diagram of their structure is on the right. Basically, these are organic chemicals that are very similar in structure. The one on the top is a dioxin. It has an extra ether bridge on the bottom. That's the only difference between it and a furan. And the only real material difference that you can know is that furans are slightly less toxic on average than dioxins, but they behave similarly. They're similarly bad for you. Um, they form naturally in the environment during combustion reactions in the presence of chlorine. So volcanoes, pretty much. Volcanoes produce them naturally. But they're also man-made by accident as impurities during chemical synthesis of chlorinated compounds. These don't have a purpose. They're not like PCBs where they have a thing that they're useful for, hypothetically, despite the problems that they cause. These are something that are made incidentally while making other things that are chlorinated. So during the Agent Orange production, during Vietnam, that was a chlorinated pesticide. A lot of pesticides were chlorinated, actually, and a lot of them are contaminated with dioxin and furan because of that. So another terminology jargon thing I need to explain. So there are differences in dioxins and furans based chemical, not so much chemical differences. They all behave fairly similarly. They're all, organic, they're all non-soluble in water, they all tend to partition to fatty materials and to sediments in the environment. Um, but their toxicity level varies depending on the number of chlorine molecules around the outside of them, as well as their positioning on the molecule. And the different permutations of where the chlorines are and how many numbers are, that's what a congener is. So if you hear me say the word congener, it's just different permutation of dioxin. Um, but they're all called dioxins and furans. But you'll see numbers before the name of the dioxin or the furan, and that number denotes where the chlorine molecules are. It's really straightforward. Um, but they're all hydrophobic, as I said. They're all non-soluble in water. They're resistant to degradation in the environment by heat, light, etc. at environmental levels, and they tend to accumulate in fatty tissues of living things because they're non-soluble. And this is a problem because using the Passaic River as an example, if you've got the sediment on the bottom of the river that's contaminated with this stuff, you got worms in the sediment, they get exposed to it, they accumulate in their tissue, fish eat the worms, other fish eat those fish, and it moves up the food chain and bioaccumulates. And the result, and obviously when we eat that fish, we get exposed to it and it builds up in our tissues. And it's problematic because these things are both acutely toxic as well as carcinogenic with long-term exposure. Particularly this arrangement of chlorine molecules. So 2,3,7,8 means that there's 4 chlorine molecules in the 2,3,7,8 position. So if you look at the structure on the right, you can see it means this type of parallel structure where you've got 2 chlorine molecules on each end. And the reason why that's a problem is because in our bodies, we have our cells are capable of mistaking this for other things because it fits into existing trying to think of a way to explain this simply. Existing structures inside of your cells, even though they're not supposed to be there. And because of that, it's absorbed into your system and can readily hurt you. Um, and this particular arrangement has actually, to give you an idea of the toxicity, my favorite fun fact about it was used to poison the tea of the Ukrainian prime minister back in 2006, I want to say, and he almost died just from having like a little vial dumped in his tea. That's how acutely poisonous this stuff is. We have to be really careful when we work with it. They can be generated directly by accident. Again, none of these are never made uh, intentionally. But they can also be formed by what's called dechlorination of more highly chlorinated dioxins and furans. So dechlorination is exactly what it sounds like on the tin. It's when you have a molecule that's got chlorine molecules around the outside, and bacteria strip off a chlorine molecule and replace it with a hydrogen molecule, and it changes the structure over time. So it's still the same basic structure, just the properties are a little bit different. And that's the whole topic of today's talk, which I'll get into more detail. But the reason why this 2,3,7,8 TCDD, TCDF is a problem for these 2 sites I work on specifically is because they are major contaminants at both sites. So TCDD, which is more toxic. Remember, I said dioxins are always a little bit more toxic than furans. Um, it is the major contaminant at the Passaic River. So PR is Passaic River, so it's New Jersey. And it's less present at the TR and SR, so it's the Tittabawassee River slash Saginaw River. I'll just use TR a lot in this paper. Um, but basically, it's present at both. However, TCDF, the furan, is actually a major contaminant at both sites. So the focus of my research the past 2 years has actually been on TCDF specifically, the furan variant of this, because it is at both sites. So to give an idea of the how bacteria can change these things by stripping off the chlorine molecules, because when you strip off chlorine molecule, you've changed the structure, right? So if you imagine, excuse me, your cells as puzzle pieces, and the chemicals as puzzle pieces, if you change the shape of the puzzle piece edge, it's not going to be able to interact with your body as easily. So if you can strip off one chlorine molecule, the EPA considers it remediated. Even if it's still there, it's still dioxin, it's still horrible for you. The degree of toxicity is so much less if you can change that into a trichlorinated dibenzodioxin or furan. It's considered done, pretty much, because the exposure is so much less risky. And the process by which bacteria do this is a special type of metabolism by specifically what are called organohalide respiring bacteria. And to make a long story short, they use hydrogen that's created during anaerobic degradation in the environment. So I know some people here are digestion people. This is a process that occurs in parallel with anaerobic digestion and anaerobic breakdown. So if you look at this picture on the right, the H2 that's some of it that's used for methanogenesis gets diverted into a different pathway that's used by organohalide respiring bacteria for dehalogenesis. Um, and the result of this is, like I said, you can detoxify these solutions in the environment. And this whole reaction was only discovered within like the last 20 years or so, so it's still a very novel topic. The problem with it is, because good, it means that, eventually the dioxin should be broken down and should be degraded in the environment. The problem is the time scale of it. It's very, very, very slow. So this is from a paper that our lab published before I started. Um, so if you look at that x-axis uh, you can see only 10% of the moles of TCDD broke down, or this was started with a penta. So it started with a penta, but you can see it's a very slow reaction. Like we're talking like not quite geological timescales, but not on a practical level for human health. We want to get these out of the environment quick. We don't want to leave these lingering contamination problems for future generations because it's already been left to us, right, by our grandparents' generation, and we don't want this to continue into the future. So my work is basically trying to figure out, okay, what do I have to feed these bacteria? What do I have to give them? What do I have to like, provide them emotional support for to get them to work faster? Get them to work faster, eat this up quicker, and get rid of this stuff so that we make it safer for the people in these majorly populated communities. So these are some of the bacteria that are capable of doing this. I just want to make it clear with this slide that it's not one specific bacteria that we're targeting. There are a few that are well known in this sphere. Dihalococoides mccartyi is the one that really started out the understanding this was even possible within like the last 20 years or so. But all of these bacteria are capable of dehalogen de um, dehalogenating some of these compounds. And where you see electron receptors, that's the compounds that they're capable of breaking down. So it's not just dioxins and furans. They're capable of breaking down like all sorts of chlorinated contaminants. So TCE, that's the dry cleaning fluid that I told you about before. Dichlorobenzene. They love dichlorobenzene. I feed them that all the time. Um, but understanding these communities, how they work, and what they need to do this is still kind of largely unknown. We're still kind of teasing that out. But when it's successful, this is what it looks like. You start with a highly chlorinated compound, and then very slowly it strips away the chlorines over time. And it can happen in different patterns. Some bacteria are better with breaking down different structures of chlorine molecules than others. Um, So you can see here the different McCartii CBDB1 can do can break this penta down to this structure, um, but the different strain 195 can break it down a different way. So it's all semantics, but if we can get a chlorine molecule off, I don't care which pathway it happens in. It's good. So, getting into how we actually stimulate these bacteria and how we measure that we're actually, successfully feeding them and getting them to work. We do what are called microcosm experiments, which are literally just little bottles that we fill with sediments from a contaminated river, a nutrient solution to give them enough food and give them a favorable environment. And then we dose them with all sorts of experimental things that we think they might like. So we start at the river, we collect this muddy, beautiful, I think Donna called it black mayonnaise slurry we usually wait for the tide to go out, and then we collect it from the surface. So it has to be, these are all anaerobic bacteria. I should have mentioned that earlier. So we have to make sure that we do this at low tide and get sediment really from the river bottom that's normally covered by water, because that's where the anaerobic species are going to live. We store them in glass jars because glass is less permeable to oxygen than plastic is. And they can sit in the fridge for years once they're packaged up like this. And then on the right is our microcosm batches, where you can see my lovely lab bench here. But it's just little serum bottles, like maybe 160 milliliters. We have some smaller ones too, which I'll talk about today, but literally like this big. But we have like hundreds of them. So once we get to the point where we have those microcosms, how do we actually analyze them? Well, we do two things. We monitor the gas portion above the liquid slurry, as well as the slurry itself, where the sediment that we collected from the river is, where the bacteria is doing its job. Um, so the headspace we measure on a gas chromatography flame ionization detector, is GCFID, and it's a fancy way of saying it sets the gas on fire and measures the chemicals inside based on the flame that comes out. We use that for measuring methane that they're producing. 'Cause remember what I mentioned before, that these do a chemical reaction that runs in parallel to anaerobic digestion. So we can actually monitor the health of the anaerobic bacteria community by seeing if they're making methane. Because even though making methane is not the goal like it is in a digestion reaction, we want to make sure that we didn't kill them, That this is how we can tell, like, okay, they're growing well, they might eat more. If we see the methane production level off, that means we need to give them more food. It's basically just a way to monitor their health. We can also measure if they're breaking down volatile organic material. So benzene is volatile, so which means that it evaporates out of the solution. So if we see like chlorinated benzenes go down and normal benzene go up in the headspace gas, that means that they're breaking it down successfully. But the slurry is where kind of the meat and potatoes of our work is. So that's where the dioxins and furans remain. Because remember, these things don't evaporate out, they don't dissolve into the water, they stay stuck to the sediment. So we analyze dioxin and furan samples on a mass spectrometer because that's the only way we can do it. We also measure volatile fatty acids with a HPLC, and we also send samples of the bacteria present out for genetic sequencing at an external laboratory. And this is something that we've only started to do recently, but I'll show you some of the exciting results we got literally like a week ago from that. That's later on. So to sum it all up, what are we trying to achieve with this? One, we're trying to make sure that this dehalogenation is happening, that, we're not wasting our time. Figure out what's happening, what's the basic rate at which it's happening, and figure out ways to stimulate the bacterial population. If we can see that it is happening, number two is, okay, which bacteria are actually driving this? Because that also, once we know which bacteria are present, we know obviously which conditions they like. So therefore, we can find better ways to stimulate it. Um, and we're also trying to figure out there's what differences are present between the rivers in New Jersey and Michigan. And overall, then the why is just better understand the mechanics of dehalogenation so that eventually we can scale up whatever treatment we're feeding them for remediating, that 17 miles of contaminated river in New Jersey. That's the dream. So the first experiment that we're doing, we took sediments from the Tittabawassee River, so it's the Michigan River, and this was kind of like a preliminary thing to see, okay, can we get them to eat this stuff? Because there's been research done on TCDD specifically being broken down anaerobically, but there's almost nothing on TCDF. So we were kind of shooting in the dark here. So we're like, all right, let's just make up 8 microcosms. Uh, we do 2 kill controls, which means that we kill the bacteria present. It's just to show that, whatever breakdown's happening is from the bacteria and not from some quality of the river sediment that we started with. Uh, we also did 3 with TCDF by itself, and then 3 with TCDF, and then a little spike of 1,2-dichlorobenzene because we knew from past work that the bacteria like dichlorobenzene. So this is kind of like a mild stimulant to get them working so that, hypothetically, if they get working eating that, hopefully they'll start eating the TCDF. Um, and then this has been running for about 18 months now. The sediments that we're using were sent to us from a lab in Michigan, and it was just literally out of some guy's refrigerator. He was like, "Yeah, it should be fine." Um, Unfortunately, I didn't even bother putting the TCDF data in here because we got nothing after a year of it sitting in the closet. No breakdown of the TCDF. So we did see on the left, you can see that we did get some methane production, which is good. Um, but the methane so it showed the bacteria was still alive, right? Which is good, but it might not have been the species that we wanted or needed for dehalogenation. Um, on the right, we did see that the dichlorobenzene broke down, which is good. So there's something in there that was capable of breaking down chlorinated organic material, but just not the stuff we're working with. So bit of a wash, but we at least had a plan. Okay, so we know that it breaks down DCB. We know that the bacteria there, whatever's in that river, seems to like it. So we gave a call to one of our collaborators in Michigan, asked them very nicely if they could send us some fresh sediment, which they did. So that's what we did for experiment 2. So this is the Tittabawassee River again, and it's what we're calling our spatial series data, which is going to be the bulk of what I'm talking about today because it's got the best data. Um, Basically, we asked our collaborator to collect sediments for us from a range of sites along the river. And he did. He very kindly, out of the goodness of his heart, went along the entire river and collected sediments from 8 sites, mailed it all to me here in New Jersey, and I made up a set of 48 microcosms. So each site has 3 kill controls and 3 bottles of just sediment and dichlorobenzene. And the reason why we didn't do furan with this is because furan is really expensive. So um, for the previous study, experiment 1, for perspective, it was, I believe, $600 for 1 milligram of the stuff. So, times are tough. Gotta pick the economic decision. Dichlorobenzene is way cheaper. So we figured, okay, this is gonna be just sort of teasing out what's there, what bacteria are present, and how are they breaking this down. So we're using dichlorobenzene as a model for TCDD and TCDF in this study. And the reason why we do 1,2 chlorination specifically is because you can see the 2 chlorine molecules are next to each other. That's the same structure that you see on the TCDD and TCDF, right? So hypothetically, since this puzzle piece is the same shape as on the side of the TCDD and TCDF, the bacteria that can break this down should be able to handle those compounds as well. So it's kind of like an indirect measure of can they do it? This one is currently at about 6 months incubation. Um, these are the sites that Dr. Upham was very kind enough to go up and down the river and get us for us. He started down here in Saginaw, which is at the bottom right, and he just worked his way up the river uh, up to Midland, which is where the hot spot is. So I guess the numbering is a little bit weird because it's in the order he collected them rather than in distance from the site. But just keep in mind, sites 1 through 4 and the data are way downstream near an urban area, and sites 5, 6, 7, 8 are further upstream, closer to the hotspot. So this one, we had great methane growth on all of them except from site 6. So that was a good sign. That means, there's some anaerobic bacteria in there that survived the overnight shipment from Michigan. But differences were apparent right away in the activity level. So this was kind of interesting because it's like, okay, so there's bacteria there, but they're all working at different rates. So that indicates that there's some difference in either the favorability of the conditions for anaerobic bacteria growth or that there's different populations of anaerobic species present. Some of them might be methanogenic, some of them might not. Some of them might just not have a lot of cells, even if the same species are present. If you're starting with a site that has less bacteria than another, Obviously, you're going to have a lot slower growth, a lot slower breakdown. And that translated into some differences that we observed in dehalogenation of the chlorinated benzenes at the site. So we started with dichlorobenzene, which is the black circle lines. These are the first four sites. These are the downstream sites. So site one, site two, site three, site four, all of them were able to break down dichlorobenzene to chlorobenzene. So that is, remember that structure before with the benzene ring with the two chlorine molecules? That means one chlorine molecule was taken away successfully. But what was really interesting was you can see at some sites, like site two and site three and site four, oopsies, they actually kept going and dechlorinated all the way just down to regular benzene. And that was really unusual because typically in past research we had seen from this laboratory and others, they're really good at taking one chlorine molecule off. And remember, like I said, EPA thinks one chlorine molecule is perfect. That means you're gravy. You did it. Congratulations. But the fact I was able to get them to take off both was huge because that hasn't really been observed much in the literature. And it means that the there's the potential there for a higher degree of dechlorination, dehalogenation. Um, but this is very poorly understood right now. I think there's only four papers in my undergrad, and I've been able to find demonstrating this so far. So we're still trying to tease out, okay, how did we do this? But it's a great sign. It's a great sign. One thing to keep in mind too, as you see these results charts, you'll see there's like little spikes of dichlorobenzene. That means that we fed more of it that week. So we do have to periodically re-amend these with more food for them, which makes sense, you know. They get hungry too. So um, that's just weeks where we fed them more dichlorobenzene. That's why you're seeing the dichlorobenzene kind of go up and down some weeks. Um, but the chlorobenzene and benzene are pretty consistently going up. One thing to keep in mind though, with these graphs is they're a time series. So if you look at site 3 in the bottom left, as dichlorobenzene goes down, chlorobenzene goes up, which makes sense, it's being transformed into that. But then you'll see as benzene goes up, chlorobenzene goes down. And that's proof that it's being transformed into benzene. So something to keep in mind as you look at these graphs. But what was really interesting was as we went upstream, we didn't see this effect as often. So if you look at site 5 and site 7, excuse me, had that full breakdown to benzene, that really unusual activity that was really promising. Site 6 had nothing. I couldn't get them to eat anything. That it even now it's just the dichlorobenzene keeps going up and up and up with each feeding, and we're not sure why. This indicates that either the bacteria is not present at all, even though they were all collected in the same method, just surface sediments from the bottom of the river, or it could mean that the conditions just aren't as favorable at that site for these dehalogenating bacteria, which is interesting. And then finally, site 8, the site closest to the hotspot in Midland, the dechlorination is stopped at chlorobenzene. We're not getting any benzene breakdown at all, or any breakdown to benzene. It's all stalled at chlorobenzene. So what the question then becomes, what's so different about these sites that's giving us these huge difference in results? And why are some sites stopping at one metabolite or another, or not even starting dehalogenation? So our next step, which we just got the data back from like a week ago, so pardon the fact that this is screenshots for now, is that there are differences present in the bacterial communities at each site. So the at the top figure here, I know, pardon the, again, that it's a screenshot, but the two samples in the middle, 7Na is site 7 pre-incubation, and then site 7 live 1 is site 7 post-incubation. Likewise, Na1, pre-incubation for site 1, live 1, site 1, post-incubation. We saw that there was differences in the dehalogenating bacteria present. So this is Dihalococoidea, which is the taxa that we normally associate with breaking down chlorinated organics. The baseline levels of these were different. Again, I know the numbers are very tiny, but you can see very clearly that the bandwidths are very different between the two sites. And then down here in the bottom graph, this one's a little bit harder to see, but site 7 actually has a lot of Dehalobacter present, which was kind of confusing. So it's little differences like that are saying, okay, that are telling us, okay, there's actually differences in the bacterial population on a spatial series, a spatial scale on this river. So, and obviously they're behaving differently in response to these organics. So then what does this say about cleaning up this river? It means that we probably can't use the same remediation approach across the whole 25 miles of contaminated river. And that's not something that the US is, NIH is going to like to hear because, they want a solution that's, you know, one plan for that'll work for the whole river. But our research is showing that that might not work. They're going to probably have to tailor the treatment to a smaller scale, and figuring out what drives those changes in bioremediation rate is kind of going to be the next step of our research on the Tittabawassee River. Now, while that's all going on, we're also running experiments on the Passaic River. So the Passaic River has been a bit more studied for this type of work because Donna is here, Dr. Fennell is here in New Jersey, obviously. So she's published some papers on breaking down TCDD on the river before, um, but not TCDF. So we took sediments from the Passaic River in summer 2024. We set up a set of microcosms, and this one's kind of interesting because it's one site, so all the sediments from one spot on the river, but we're testing it with a bunch of different amendments. So to see if, some of them work better or others. So the co-amendments that we're testing here are cobalamin, which is vitamin B12, because dehalogenating bacteria like vitamin B12. It's a limiting thing for them. Also, we're testing ways to provide them with more habitat to grow on by amending it with activated charcoal. We've also tried a slow release electron donors, that's just their food. Um, because a lot of commercial products for bioremediation now in the environment are moving towards these slow release electron donors for groundwater. So we thought, why not? Let's try it. They gave us a free sample with this company, so um, bacteria seem to like it. They like they're putting out a lot of methane after being fed that. And again, the dichlorobenzene, because that's kind of the link that ties this research into the spatial series data. This is on month 12 of incubation. I'm going to be presenting data up to month 6 because we're still analyzing data from this. But so far, as far as the data that we've analyzed, we're getting really, really good bacterial growth. There's a lot of organics in the Passaic River. I actually haven't had to feed them any new volatile. We feed them a volatile fatty acid solution, like acetic acid and things like that, to keep them growing. I haven't had to feed them once since we started this experiment because they are putting out so much methane. Um, but again, we're seeing some differences in methane growth, so in bacterial growth based on the treatment that we're giving them. So that's a good sign. So on the right, you can see all the permutations that we're testing. And the data from this, like I said, we're still analyzing it, but hopefully we'll see this driving some differences in the dechlorination rate of TCDF. Hopefully we'll see TCDF breakdown this time after the failure of the Tittabawassee River. So fingers crossed there. So we're also continuing some experiments from my predecessors in the lab. Rachel Dean, she published the paper that I mentioned before on TCDD breakdown in the Passaic River with dichlorobenzene. Her microcosms are about 5 years of incubation, so I've been just taking care of them, feeding them, continuing to measure them. Likewise with some of Donna's other students, JC Miranda and Eric Nitzberg. We've got about 40 microcosms between the two of them, testing all sorts of different congeners of dioxins and sacred sediments. And hopefully we can use these to make enrichments. So that's like, highly cultivated bacterial cultures that we can use for bioremediation in the future. So our conclusions and new questions and all this is raised. Uh, I've gotten really good at breaking down dichlorobenzene. Not so much the TCDF, which is our goal, but I have demonstrated that these dehalogenating bacteria are there. They can do it. Why they're not eating the TCDF, we're not sure, but we're working on that. Dehalobacter seems to be a pretty common taxa at the most productive sites, at least for the DCB breakdown, but we're not sure what's driving the differences in the population growth between sites. That's going to require a lot more genetic analysis in the coming weeks, which hopefully we have funding for. Um, and as I mentioned, one size won't fit all for these treatments on these rivers. It seems like they're going to have to do a lot more in-depth site analysis of like breaking the river into chunks and thinking, okay, what treatment can we tailor that best suits the bacterial community here? And what can we, so we're also going through a lot of like historical data of various people who have done genetic work at these sites. That we're going to plan to do that. Pick MSU's brain and see how much data they have from past work on this river. And how can that change over time too? 'Cause remember, I mentioned the Midland floods that are so common on the Tittabawassee. How will that change this bacteria population over time? Because obviously, you would expect things to get shifted around and move. A river is not a static system. So will those treatments also be effective over a time scale as well? And that's kind of a question that we're trying to answer. So some future plans, as I mentioned, we're trying to scale up the spatial theories cultures because they seem to be really doing great. Um, I'd like to start microcosms with enriched cultures from those sites or dosing some of our old cultures with enrichments from them. Do more genetic data, obviously, or get more genetic data. Um, and then the pie in the sky dream would be to figure out, is there something new in there that's, driving this difference that we're observing? Figuring out if we could do that. Um, Again, not very feasible or likely, but that would be the dream to prove that we found something new, something novel. And then just to end things, I wanted to bring this back to the food, water, energy nexus because I was part of the stewards, and I know this is a big focus of the work there that we did. Um, but dioxins, to give you a better picture of how they've actually hurt people on a practical scale in the environment, there's been a lot of documented, again, this is a worldwide problem. These are a few cases that I found after some light Googling on the NIH website and on Web of Science. There's been widespread documentation of contaminated fish in the Baltic Sea. So that feeds the Nordic countries, Russia, that whole region is being impacted. Likewise, in Italy, they found that it was contaminated by the local fish were contaminated by feed that had a chloride molecule, it's a chlorine containing compound that happened to be contaminated with dioxin and furan. And now, and once you find dioxin in a site, usually the government will issue a recommendation not to eat it. Like for New York, you can't eat, you're not supposed to eat fish out of the entire New York harbor or Newark harbor because of the dioxin contamination and the risk of exposing people to it. And then perhaps most importantly of all the sites I've talked about is in Vietnam, where because of the Agent Orange that was produced in New Jersey and elsewhere in the US, um, this is where it was actually used as a weapon. So as a result, the residues are still present in the soil and the water, and the whole country has massive issues with contaminated sites of canals, soils. It's showing up in the fatty tissue of livestock, geese in particular. And this, again, once you find that contamination, the people have lost a source of food, of safe food. So then it becomes, okay, do you eat this and starve, or do you eat this and not starve? But then you're risking exposure to this. So it becomes an environmental justice issue in that point. Because if it's the only food people have, you know, can you tell them not to eat it? And just to really emphasize, I found this so funny in a sad way. These are some news articles that recently came out. The one on the left is from Newark, where the city of Newark, where the Passaic River contamination is from the manufacturer of Agent Orange. This is causing a lot of stress in the community because the river can't be used for recreation, for food, for anything really, aside from just sitting there. So the communities there have no way of using it effectively because of this contamination. And on the right, you see the communities that are hit in Vietnam because they also can't access their waterways or use them or anything like that. So it's kind of an irony that both the places where these weapons were made and where they were used are unable to use their waterways because of this contamination. This is a worldwide problem. And cleaning these up in the future is being thrown into question because, like so much in science, it's very funding dependent. And that article on the right was talking about how USAID was actually driving a lot of the cleanup efforts in Vietnam for the dioxin contamination. So it's unclear at this point if that will continue without finding external funding. But it really emphasizes why it's so important to find cheap and affordable methods of stimulating the bacteria that are present to actually do this natural process because that might be the only option that we have if this funding keeps getting cut. So not to end on a downer note, but uh, just some acknowledgments. I'd like to thank the co-PIs at Rutgers that work with me. It's Dr. Donna Fennell, as I know, is on the call. Gerben Zylstra, our microbiologist, who has been a huge help because I'm not a microbiologist in interpreting the genetic data we've been getting. our PhD student and undergrad students, as well as all of our collaborators at Michigan State. Our funding for this came from the NIH uh, through the Superfund research program. It's been running since the 90s, I believe, which funds research into novel methods of cleaning up these contaminated sites. So, any questions?